Everybody in the longevity world is talking about mitochondria, the cellular powerhouse, the energy factories, NAD plus supplements, red light therapy, cold plunges. And look, those interventions work. The research is solid, but here's the thing nobody is saying out loud. You don't have to spend hundreds of dollars on supplements or sit in an ice bath to restore mitochondrial function. There are specific compounds found in ordinary food, compounds with documented, peer-reviewed, mechanistic evidence that activate the same molecular pathways that expensive interventions activate, without the cost, without the ice water shock, without adding 15 different protocols to your already complicated day. I'm not talking about supplements from an Instagram influencer with a discount code. I'm talking about 5 specific foods and compounds, one of which you've probably never heard of despite being studied for decades, and one of which is hiding in vegetables you walk past every single week, that the research now shows can meaningfully restore mitochondrial function at the cellular level. Compound number 4, specifically, I want you to wait for it, because the research behind it is extraordinary. The mechanism is one of the more elegant things I've come across in cellular biology, and it's available at any grocery store for less than the price of a single supplement bottle. Let me show you all 5, what they contain, what that compound actually does at the mitochondrial level, how much of each the research suggests you actually need, and why after age 40, this becomes increasingly non-negotiable if you want to maintain energy, cognitive function, and metabolic health. Before we get into the compounds, I want to make sure we're on the same page about what mitochondrial decline actually looks like after 40. Because the phrase gets used casually in a way that strips away what makes it genuinely dangerous. Mitochondria are the organelles responsible for converting the food you eat and the oxygen you breathe into ATP, adenosine triphosphate, the energy currency that powers every single cellular process in your body. When you move your muscles, that's ATP. When your heart beats, that's ATP. When neurons fire to form thoughts and memories, that's ATP. When your immune system mounts a defense against pathogens, that's ATP. But here's what happens around age 40, and the research on this is clear. Mitochondrial DNA begins accumulating mutations and oxidative damage at an accelerated rate. By middle age, mitochondrial DNA volume, integrity, and functionality decrease significantly. A study published in PNAS examined skeletal muscle mitochondrial function across 146 healthy adults aged 18 to 89. And the findings were striking. Mitochondrial DNA abundance, mitochondrial ATP production, and aerobic capacity all declined progressively with age. This isn't a gradual slowdown. This is a structural breakdown of the cellular machinery that determines whether you wake up with energy or drag through the day on caffeine and willpower. With advanced age, you see impaired oxidative capacity, reduced oxidative phosphorylation, decreased ATP production, significant increases in reactive oxygen species generation, and diminished antioxidant defense. But here's where it gets interesting. The decline isn't uniform across all tissues. In heart muscle, particularly interfibrillar mitochondria, complexes 3 and 4 lose activity, while in skeletal muscle, liver, and brain, complex 1 appears more sensitive to age-associated dysfunction. Complex 1 is the largest respiratory chain complex, and it's ground zero for age-related mitochondrial failure. When complex 1 starts malfunctioning, ATP production drops, and cells shift into a metabolic state that increases inflammation, insulin resistance, and cellular senescence. Now, why does this matter specifically after 40? By age 40, NAD+ levels, the coenzyme that fuels over 500 enzymatic reactions in your body, drop by approximately 50% compared to youthful levels. By age 60, the decline can reach 80% or more. The major culprit is an enzyme called CD38, whose levels rise during aging. CD38 is an NADase, meaning it breaks down NAD+, and as CD38 activity increases with age, NAD+ availability plummets. Stay with me because this cascade is critical. NAD+ is required for the function of sirtuins, a family of proteins that regulate mitochondrial function, DNA repair, inflammation, and cellular stress responses. SIRT3, the main mitochondrial deacetylase, plays a unique role in maintaining mitochondrial health, and loss of SIRT3 function has been linked to insulin resistance, cancer, cardiovascular diseases, and neurodegeneration. When NAD+ levels drop, sirtuins can't function properly. When sirtuins can't function, mitochondria can't maintain quality control. When mitochondrial quality control fails, damaged mitochondria accumulate, oxidative stress escalates, and cellular aging accelerates. Altered mitochondrial dynamics with chronological age can inhibit mitophagy, the process that removes dysfunctional mitochondria, leading to accumulation of damaged organelles in cells. This is where aging becomes cellular. This is where fatigue, brain fog, metabolic inflexibility, and disease risk aren't just symptoms of getting older, but direct consequences of mitochondrial dysfunction. And this is exactly where targeted nutritional compounds can intervene. Let's talk about compound number 1, pyrroloquinoline, PQQ. You've probably never heard of it. The supplement industry hasn't latched onto it the way they have CoQ10 or resveratrol, but the research on PQQ and mitochondrial biogenesis is some of the most compelling in nutritional science. Mitochondrial biogenesis is the process by which cells generate new mitochondria. It's not just maintaining the ones you have, it's building fresh, functional mitochondria to replace damaged ones. When researchers exposed mouse hepatocytes to PQQ for 24 to 48 hours, they observed increased citrate synthase activity, increased cytochrome C oxidase activity, increased mitochondrial DNA content, and increased cellular oxygen respiration. Those aren't abstract markers. Citrate synthase is a key enzyme in the citric acid cycle. Cytochrome C oxidase is complex 4 of the electron transport chain. When both increase, you're looking at enhanced ATP production capacity. But here's where the mechanism gets interesting. PQQ works by activating CAMP response element binding protein, or CREB, and peroxisome proliferator activated receptor gamma, coactivator 1-alpha, PGC1-alpha, a pathway known to regulate mitochondrial biogenesis. PGC1-alpha is called the master regulator of mitochondrial biogenesis. When PGC1-alpha gets activated, it triggers a cascade of transcriptional events that increase the expression of nuclear respiratory factors, NRF1 and NRF2, which in turn upregulate mitochondrial transcription factor A, or TFAM, and other genes that control mitochondrial DNA replication and respiratory function. When researchers knock down either PGC1-alpha or CREB expression using small interfering RNA, PQQ could no longer stimulate mitochondrial biogenesis, confirming that PQQ's effects are mediated through this specific pathway. Now this is where it gets practical. A randomized controlled trial of 140 adults, aged 40 to 65, published in the American Journal of Clinical Nutrition, demonstrated that 1,000 milligrams of NMN daily increased blood NAD plus levels by an average of 38% after 60 days. But with PQQ, the dosing is different. In human subjects, a single dose of PQQ at 0.3 milligrams per kilogram of body weight, approximately 20 milligrams for an average person, produced a definitive increase in mitochondrial function as measured by urinary metabolites within 48 hours. In one study, 41 healthy elderly subjects were randomized to receive either 20 milligrams of PQQ daily for 12 weeks or a placebo. Compared to placebo, PQQ supplementation was associated with significant improvements in attention and working memory, and an associated increase in brain blood flow in the frontal lobes. A study on untrained men showed that while PQQ supplementation at 20 milligrams per day did not produce ergogenic effects on aerobic performance, it did significantly increase PGC1 alpha protein levels, the biochemical marker for mitochondrial biogenesis, compared to placebo after 6 weeks of endurance training. So PQQ doesn't just maintain mitochondria, it actively stimulates the creation of new mitochondria through a well-characterized molecular pathway. Food sources of PQQ include fermented soybeans, natto in particular, green peppers, parsley, kiwi, and papayas. But the concentrations in food are extremely low, typically in the range of nanograms per gram. Levels of PQQ from dietary intake from plants are sufficient to maintain the concentration of PQQ typical of tissues. But for therapeutic effect, supplementation at 10 to 20 milligrams appears necessary based on the human trials. The second compound is one you already know, but you probably don't understand its mitochondrial mechanism, coenzyme Q10, specifically in its reduced form, ubiquinol. CoQ10 sits at the center of the electron transport chain. It shuttles electrons from complex 1 and complex 2 to complex 3, enabling the proton gradient that drives ATP synthesis. Complex 1 is essential. If it doesn't function properly, no energy can be generated. Complex 1 is most affected by aging and is the primary site where free radicals are produced. Here's the elegant part. Ubiquinol enables the electron transport process to skip complex 1 and complex 2 and proceed directly to complex 3 to produce ATP. With ubiquinol, more protons are pumped, enhancing cellular energy generation while reducing free radical production. This is critical after 40 because endogenous CoQ10 biosynthesis and tissue concentrations decrease with age, with reported reductions in heart, skeletal muscle, and other tissues. But it's not just about maintaining baseline levels. In a study on epidermis tissue, researchers observed a decrease in mitochondrial respiration and ATP production with donor age. However, when the reduced form of CoQ10, ubiquinol, was administered, there was a regeneration of mitochondrial respiratory parameters. Ubiquinol supplementation in senescence-accelerated mice enhanced mitochondrial activity by increasing levels of SIRT1, PGC1 alpha, and SIRT3, slowing the rate of age-related hearing loss and protecting against the progression of aging and symptoms of age-related diseases. And the mechanism goes beyond electron transport. Ubiquinol may activate SIRT1 and PGC1 alpha by increasing cyclic adenosine monophosphate, or CAMP, levels that in turn activate CREB and AMPK. AMPK is the cellular energy sensor. When AMPK gets activated, it shifts metabolism toward fat oxidation, enhances mitochondrial biogenesis, and improves insulin sensitivity. Moreover, ubiquinol detoxifies superoxide anion radicals and peroxyl radicals and regenerates alpha tocopherol, vitamin E. This matters because oxidative stress inside mitochondria creates a vicious cycle. Reactive oxygen species damage mitochondrial DNA. Damaged mitochondrial DNA produces dysfunctional respiratory complexes. Dysfunctional complexes generate more reactive oxygen species. Ubiquinol breaks that cycle by acting as both an electron carrier and an antioxidant within the mitochondrial membrane. Dosing matters here. In cellular studies, CoQ10 supplementation at concentrations of 100 micromolar positively correlated with improved mitochondrial functionality, including increased oxygen consumption rate, transmembrane potential, and ATP synthesis. In humans, doses of 100 to 300 milligrams of ubiquinol daily appear in most clinical trials, showing cardiovascular and metabolic benefits. Food sources include fatty fish like salmon and mackerel, organ meats, particularly heart and liver, and certain vegetables like spinach and broccoli. But again, the concentrations are low. A 100-gram serving of beef heart contains roughly 11 milligrams of CoQ10, so dietary intake alone won't reach therapeutic levels for someone over 40 with declining endogenous synthesis. Now let's talk about compound number 3. And this is where the research on cellular stress response gets fascinating: sulforaphane. Sulforaphane is an isothiocyanate derived from cruciferous vegetables, broccoli, Brussels sprouts, kale, cauliflower, and it operates through a completely different pathway than PQQ or CoQ10. Sulforaphane activates nuclear factor erythroid 2-related factor 2, or NRF2, a transcriptional factor that regulates the expression of hundreds of genes involved in antioxidant defense, detoxification, and mitochondrial function. Here's how it works mechanistically. Under normal conditions, NRF2 is bound to a protein called KEAP1 in the cytoplasm and rapidly degraded. Sulforaphane stabilizes cytoplasmic NRF2 through KEAP1 binding to inhibit NRF2 ubiquitination. When NRF2 stabilizes, it translocates to the nucleus and binds to antioxidant response elements, or AREs, in the DNA, activating the transcription of protective genes. Stabilized NRF2 exerts transcriptional activation of ARE responsive genes involved in redox regulation, fatty acid metabolism, glucose metabolism, and amino acid catabolism, including enzymes and cellular transporters. This results in provision of metabolic substrates for mitochondrial machinery, the tricarboxylic acid cycle, fatty acid beta oxidation, and oxidative phosphorylation complexes, as well as mitochondrial transporters. But NRF2 doesn't just prepare fuel for mitochondria. NRF2 may also directly upregulate ARE-bearing genes that encode mitochondrial apparatus and mitochondrial quality control processes, including mitophagy and biogenesis. The research on this is striking. In a study using free fatty acid-treated cells and high-fat diet-treated rats, researchers observed that sulforaphane alleviated the swelling of mitochondria and stimulated mitochondrial biogenesis. The reduced expression of NRF1 and TFAM was reversed by sulforaphane. NRF1, nuclear respiratory factor 1, is a transcription factor that regulates the expression of genes encoding subunits of the respiratory complexes. TFAM is the mitochondrial transcription factor that controls mitochondrial DNA replication. In senescence-accelerated mice fed sulforaphane-containing broccoli sprout powder At 0.3% weight per weight until 13 months of age, researchers found that sulforaphane significantly improved long-term memory, hippocampal mRNA, and protein levels of PGC1-alpha and TFAM, master regulators of mitochondrial biogenesis, increased in both control and senescence-accelerated mice. And here's the part that makes sulforaphane unique. In old mice, sulforaphane restored NRF2 activity, mitochondrial function, cardiac function, exercise capacity, glucose tolerance, and activation and differentiation of skeletal muscle satellite cells. This isn't just protecting existing mitochondria. This is restoring the signaling pathways that decline with age and re-engaging the cellular machinery for repair and regeneration. Sulforaphane prevented cholesterol-induced alterations in the coupling efficiency of mitochondrial respiration, improving ATP turnover and spare capacity, and averted the impairment of electron flow at complexes 1, 2, and 4. Spare capacity is the reserve energy production capacity your mitochondria hold in reserve for moments of increased demand, exercise, stress, immune activation. As mitochondria age, spare capacity erodes, and cells lose metabolic flexibility. Now, the practical question: how much sulforaphane do you need, and where do you get it? Sulforaphane doesn't exist directly in broccoli. It's formed when an enzyme called myrosinase converts a precursor compound, glucoraphanin, into sulforaphane. This happens when you chew raw cruciferous vegetables or when you chop them and let them sit. Cooking destroys myrosinase, which is why steamed broccoli produces far less sulforaphane than raw or lightly cooked broccoli. In the mouse study, broccoli sprout powder containing 0.3% sulforaphane, glucosinolate by weight, produced significant cognitive and mitochondrial benefits. In human equivalents, that translates to roughly 30 to 60 milligrams of sulforaphane daily, which you can get from approximately 100 to 150 grams of raw broccoli sprouts or 300 to 400 grams of raw or lightly steamed broccoli. Broccoli sprouts are the most concentrated source. 3-day-old broccoli sprouts contain 10 to 100 times more glucoraphanin than mature broccoli. You can grow them at home in a jar with minimal effort. You soak broccoli seeds overnight, rinse them twice a day for 3 to 5 days, and you have the most potent dietary source of sulforaphane available. Now let's move to compound number 4, and this is the one I told you to wait for: alpha-lipoic acid, also called thioctic acid or 1,2-dithiolane-3-pentanoic acid. Alpha-lipoic acid is unique because it functions both as an enzyme cofactor in mitochondria and as a multifunctional antioxidant outside of mitochondria. Alpha-lipoic acid is synthesized enzymatically in plant and animal mitochondria from octanoic acid and cysteine. It acts as a cofactor for pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase activity, and is required for the oxidative decarboxylation of pyruvate to acetyl-CoA, a critical step bridging glycolysis and the citric acid cycle. Without alpha-lipoic acid, your cells cannot efficiently convert glucose into usable energy. It sits at one of the most fundamental metabolic junctions in cellular respiration. But here's where it gets interesting for mitochondrial repair. Treatment of muscle cells with 0.3 millimolar alpha-lipoic acid triggers a significant increase in the expression of DNAJB3 mRNA and protein, a heat shock protein cochaperone, with a novel role in mitigating metabolic stress and promoting insulin signaling. Heat shock proteins are part of the cellular stress response. They refold misfolded proteins, prevent protein aggregation, and help maintain proteostasis, the balance of protein synthesis and degradation. Alpha-lipoic acid's therapeutic use in improving insulin sensitivity and promoting glucose metabolism has been well documented in both humans and animal models of obesity associated with insulin resistance and type 2 diabetes. But insulin resistance and mitochondrial dysfunction are not separate problems. They're two sides of the same metabolic breakdown. Insulin resistance is closely tied to mitochondrial dysfunction. When mitochondria are inefficient, cells shift toward incomplete glucose oxidation and lipid accumulation. This metabolic inflexibility worsens insulin resistance and increases oxidative stress. Alpha-lipoic acid enhances insulin sensitivity and glucose uptake by activating the PI3K/AKT pathway, a crucial signaling pathway for cellular glucose metabolism. It stimulates glucose uptake by increasing the activity of PI3K and AKT, which are crucial for the translocation of glucose transporters like GLUT4 to the cell membrane, mimicking the action of insulin. And here's the AMPK connection again. Alpha-lipoic acid activates AMPK in skeletal muscles. When AMPK phosphorylates and inactivates acetyl-CoA carboxylase, this results in decreases in malonyl coenzyme A, a critical precursor of fatty acid biosynthesis, and a potent inhibitor of mitochondrial fatty acid oxidation. Decreased malonyl CoA content results in reduced fatty acid synthesis and increased fatty acid oxidation. Translation: alpha-lipoic acid shifts your metabolism away from storing fat and toward burning fat for energy, which takes pressure off mitochondria and improves their efficiency. In a study of overweight and obese PCOS patients, alpha-lipoic acid administration at 400 milligrams per day for 12 weeks significantly improved insulin sensitivity and decreased liver enzyme levels in all subjects. In patients with type 2 diabetes, intravenous infusion of alpha-lipoic acid increases insulin-mediated glucose disposal, though oral administration appears less effective unless absorption is optimized. But the mitochondrial benefits extend beyond insulin sensitivity. Alpha-lipoic acid acts as a crucial cofactor of the mitochondrial alpha-ketoacid dehydrogenase complexes involved in carbohydrate metabolism. It scavenges free radicals, regenerates cellular antioxidant agents such as glutathione, vitamin C, and vitamin E, and modulates several critical signal transduction pathways. Alpha-lipoic acid quenches reactive oxygen species, chelates metal ions, and reduces the oxidized forms of other antioxidants. It also boosts antioxidant defense systems through NRF2-mediated antioxidant gene expression and by modulation of peroxisome proliferator-activated receptor-regulated genes. Wait, alpha-lipoic acid activates NRF2, the same pathway sulforaphane activates, and it activates AMPK, the same pathway CoQ10 activates, and it regenerates glutathione, vitamin C, and vitamin E, multiplying their antioxidant effects. This is what I mean by stacking mechanisms. These compounds don't just work in isolation. They reinforce each other's pathways and create synergistic effects on mitochondrial health. Food sources of alpha-lipoic acid include organ meats, particularly liver and kidney, spinach, broccoli, tomatoes, and Brussels sprouts. But again, concentrations are low. A 100-gram serving of beef liver contains roughly 1 to 3 milligrams of alpha-lipoic acid. Clinical studies showing metabolic and mitochondrial benefits use doses of 300 to 600 milligrams daily, sometimes up to 1,800 milligrams in divided doses for diabetic neuropathy. Our alpha-lipoic acid is the naturally occurring isomer and appears more bioavailable than the synthetic racemic mixture. So if you're supplementing, look for the R form. Now let's talk about the fifth compound, and this one ties everything together in a way that makes the other four more effective. Nicotinamide riboside, or NR, and nicotinamide mononucleotide, or NMN. These are NAD+ precursors. Remember the NAD+ decline I mentioned at the beginning? By age 40, NAD+ levels drop by approximately 50%. By age 60, the decline can reach 80% or more. NAD+ levels are required not only for life, but for a long life. When NAD+ levels fall, sirtuins, the deacylase enzymes that regulate mitochondrial function, DNA repair, and inflammation, lose activity. NAD+ levels steadily decline during aging. By the time a mouse or human is middle-aged, levels of NAD+ have fallen to half of youthful levels, with resulting loss of sirtuin and PARP activity. PARPs are poly-ADP ribose polymerases, enzymes involved in DNA repair. As levels of DNA damage increase with aging, PARP1 consumes and depletes NAD+. In support of this, PARP activity was shown to increase with age in mice and human male skin. PARP inhibition prevented NAD+ depletion and premature aging in a mouse model with defective DNA repair and boosted NAD+ levels, improving mitochondrial fitness and function. So aging creates a feedback loop. More DNA damage activates PARPs, which consume NAD+, which depletes the NAD+ pool available for sirtuins, which impairs mitochondrial quality control, which increases oxidative stress, which causes more DNA damage. Breaking that loop requires restoring NAD+ levels. In the case of aging, mitochondrial dysfunction occurs in parallel with NAD+ depletion in muscle tissues, often linked to muscle wasting or loss of muscle function. Treating mice with the salvage precursor nicotinamide riboside resulted in NAD+ repletion in muscle stem cells of both young and aged mice. This was mirrored by reduced muscle stem cell senescence and increased mitochondrial respiration and membrane potential, paralleled by improved fitness and exercise performance. In mouse models, NAD+ depletion in muscle led to progressive loss of muscle function, while NR treatment restored mitochondrial function and induced muscle remodeling and restoration. But here's the key. NAD+ boosting works best when mitochondria are protected from oxidative stress and when mitochondrial biogenesis is actively stimulated. If you boost NAD+, but don't address the oxidative damage with compounds like ubiquinol and alpha-lipoic acid, the NAD+ gets consumed faster by PARPs, repairing DNA. If you boost NAD+, but don't activate mitochondrial biogenesis with compounds like PQQ and sulforaphane, you're just maintaining damaged mitochondria at a higher energy state, which doesn't solve the underlying dysfunction. This is why the 5-compound stack makes sense. PQQ and sulforaphane drive mitochondrial biogenesis. CoQ10 and alpha-lipoic acid protect existing mitochondria from oxidative damage and improve metabolic efficiency. NR or NMN restores the NAD+ pool that fuels sirtuins and mitochondrial quality control. A randomized controlled trial showed that 1,000 milligrams of NMN daily increased blood NAD+ levels by an average of 38% after 60 days. Another study found that high-intensity interval training increased skeletal muscle NAD+ levels by 127% in adults aged 55 to 75 after 12 weeks. The mechanism involves exercise-induced upregulation of NAMPT, the rate-limiting enzyme in the NAD plus salvage pathway. So exercise itself boosts NAD plus production, but if you're over 40 and your NAMPT expression is declining, supplementation with NR or NMN can bypass that bottleneck. Time-restricted eating, intermittent fasting, has been shown to activate AMPK and increase NAD plus levels. A 16 to 8 fasting protocol increased hepatic NAD plus by 50% in study participants over 12 weeks. This is where lifestyle and supplementation converge. Fasting activates AMPK and boosts NAD plus. Exercise activates AMPK and boosts NAD plus. Alpha-lipoic acid activates AMPK. Sulforaphane activates NRF2, which supports mitochondrial quality control. PQQ and CoQ10 enhance mitochondrial function and biogenesis. Every intervention reinforces the others. Now let me give you the practical protocol, the exact daily regimen based on the research we've covered. Morning, ideally on an empty stomach or with a small amount of fat for absorption. 20 milligrams of PQQ, 200 to 300 milligrams of ubiquinol, 300 to 600 milligrams of R-alpha lipoic acid, 250 to 500 milligrams of nicotinamide riboside, or 500 to 1,000 milligrams of NMN midday, with or after a meal containing cruciferous vegetables, 100 to 150 grams of raw broccoli sprouts, or 300 to 400 grams of lightly steamed broccoli, chopped and allowed to sit for 10 minutes before cooking to maximize sulforaphane formation. Evening, optional second dose if you're targeting metabolic repair. Additional 300 milligrams of R-alpha lipoic acid. This isn't a supplement stack you need forever. This is a targeted intervention to restore mitochondrial function after decades of decline. You run this protocol for 8 to 12 weeks, then reassess. Markers to track: fasting glucose, fasting insulin, HOMA-IR for insulin sensitivity, HS-CRP for inflammation, VO2 max, or a simple step test for aerobic capacity, subjective energy levels, sleep quality, cognitive clarity. If you see improvements in these markers, you can scale back to a maintenance dose, perhaps half the amounts, and rely more heavily on dietary sources and lifestyle interventions like exercise and time-restricted eating. But if you're over 40 and you're experiencing unexplained fatigue, brain fog, metabolic dysfunction, or declining physical performance despite adequate sleep and nutrition, mitochondrial decline is almost certainly involved. And the research is clear. A growing body of evidence suggests that aging and age-related diseases are tightly related to an energy supply and demand imbalance, which might be alleviated by a variety of interventions, including physical activity and calorie restriction, as well as naturally occurring molecules targeting conserved longevity pathways. These 5 compounds, PQQ, ubiquinol, sulforaphane, alpha-lipoic acid, and NAD plus precursors, target those conserved longevity pathways. They're not magic. They're molecular tools that activate the same pathways your body used efficiently when you were younger. The pathways are still there. The genes are still there. The cellular machinery is still there. It's just running at reduced capacity because of accumulated oxidative damage, declining NAD+, and impaired quality control. You can restore that capacity. The research shows it. The mechanisms are understood. The dosing is established. The only question is whether you're willing to treat mitochondrial health with the same seriousness you treat cardiovascular health or metabolic health. Because at a fundamental level, they're all downstream of the same process. Your mitochondria determine how much energy you have to live your life. They determine how well your brain functions. They determine how efficiently your muscles contract. They determine how effectively your immune system responds to threats. They determine how long your cells can maintain homeostasis before they senesce or die. Aging leads to a decline in mitochondrial efficiency, marked by increased reactive oxygen species, accumulation of mitochondrial DNA mutations, impaired oxidative phosphorylation, and breakdown of mitochondrial quality control systems. Such changes are associated with the development of neurodegenerative, cardiovascular, and metabolic diseases. But that decline is not inevitable, it's not irreversible, and it doesn't require expensive interventions or unproven protocols. 5 compounds, well-researched mechanisms, established dosing, practical implementation. PQQ for mitochondrial biogenesis, ubiquinol for electron transport and antioxidant protection, sulforaphane for NRF2-mediated stress response and mitochondrial quality control, alpha-lipoic acid for metabolic flexibility and antioxidant regeneration, NAD plus precursors for sirtuin function and cellular repair. Stack them, track your markers, give it 8 to 12 weeks, and see what happens when you give your mitochondria the molecular support they need to function the way they did before age 40. This isn't about anti-aging. This is about maintaining the cellular energy systems that determine whether you thrive or merely survive.